it's me Manoj Bohara. Today we will be discussing about the complementary system, which is one types of innate immune system. In a complementary system, there are 20 protein, which is mainly produced by liver. They are inactive in our bloodstream, but active in a inflammation reaction. Then we'll discuss about the mechanism. Suppose there is one bacteria. This is bacteria, and on its surface there is one of the, this is antigen. When the antibody this is an antibody. When antibody binds with an antigen, then there is one of the outgrowth like this structure. Then only the complement system will be active and come here. At first C1 protein come and attach there, then the C4 be attached here, when it has one of the C4A also components, but only C4B component only attached to the C1. Then it comes by C2B component, then C4B plus C2B. These two protein are called as the C3B convertase, because these two proteins can be convert the C3B protein from inactive form to active form. Then C3B, then C3B protein attached here, then C4B plus C2B plus 3 C3B. These three proteins activate the C5B. These three protein called as the C5B convertase, because these three protein can activate the C3B. C2B also has C2A component and C3B also has C3A component, but only the C4B, C2B, C3B only attached to the C1. Then it attaches to the C5B protein. Then here there is one also combine the C6 protein, C7 protein, C8 protein and C9 protein. This is called as the terminal. This is called as the terminal part of complement system. Then this protein can be detected, and they can detect this is a bacteria. After the details of C5 to C9, they create a This is the terminal part of the complementary system, which can create a hole in a bacterial membrane. This is the terminal part of the complement system which can create the hole and destroy the bacteria. By this method. Then there are two types of bacteria. One is gram positive. It has a thick membrane, and another is gram negative bacteria, which has thin membrane and which has one of the outer double membrane. So this complement system easily destroy the gram negative bacteria, and the person who suffer from a liver disease, there should not be produce of the lot of the complement protein. So if the people who do not have the terminal part of the complement system, then that person should be suffer from the disease like as the gonorrhea. The bacteria mostly attack to the person who does not have the terminal part of the complementary system, that is the gonococcus and monococcus bacteria, which is a mainly sexually transmitted disease, which can be attached to the person who does not have the terminal part of the complementary system. Then we'll discuss about gram positive. This is gram positive bacteria. This is gram positive bacteria, and here is one of the. This is antigen. Then there is the combined. Then there is the attachment of antibody. And then the outgrowth. There is outgrowth. This outgrowth indicates to attract the complement protein. Then it come to the. Then at first is come to the C1 complement protein. Then C4b complement protein. Then C2b complement protein. Then C3b complement protein. And C3b complement protein. Then it can be destroyed by. There is one of. This is a neutrophils. And the C3 protein receptor is present here. There is the present of C3b. There is the present of C3b receptor, so the neutrophil can easily attach to the C3b protein. Then the neutrophils can engulf the bacteria by this process. This process is called as the ozonization. Ozonization is a process in which the one end of the complement protein is attached to the pathogen, and another end of the complement protein is attached to the neutrophils or macrophages, and they can destroy the bacteria or pathogen by this process is called as the ozonization. Thank you everyone. Like, comment and share and subscribe the channel.